Well, so we are doing LAC OPERON. It's a good example of economy in a way that, and bacteria, every gene doesn't have its own operator, right? So group of genes will be under one operator. So we call it LAC OPERON. We call it OPERON. So can you help me understand again what is OPERON? Hello, when there is group of genes under one. Let's see, this is DNA. Bacterial DNA is not linear. Just it's a part of that circular DNA, right? Oh yeah, please. Well, so this is, let's say this is one gene. There are 3 genes. We used to call it Z, Wow, it like Z, like Y. And all these 3 have one operator, Operator is, say, this place. And on the left of it, there's another region, and we call it promoter. Now, this all is OPERON. If they ask you to label, I cannot highlight it, but anyway, if they ask you to label the LAC OPERON, you highlight, so you should do this. What's OPERON? Group of genes under along with their operator and promoter. अगर हम LAC OPERON का पूरा वो करें, मतलब when we say ना, and in LAC OPERON there is a fact. What happens in LAC OPERON? That there are 3 genes under one operator, but that's not the LAC OPERON. नहीं समझ आ रही? I mean, yeah, I mean OPERON includes all what I highlighted. But what is there in OPERON? OPERON कहते हैं, बेटा, OPERON is when group of genes are under one operator. But when you say what does LAC OPERON comprise of? So we say LAC OPERON comprise of 5 things, sorry. 3 are the structural genes. Isabhai, 3 are structural genes. 1 is their operator, combined operator, and then 1 is their promoter. Sorry? No. Eukaryotic DNA is very long. Every gene will have its own operator and promoter. But अच्छा, OPERON बहुत तरह के होते हैं. LAC OPERON भी है, TRIP OPERON भी है. TRIP OPERON comes very much in papers. That is when the group of genes which deal with tryptophan. Tryptophan is an amino acid. So far, if I ask you, what is our takeaway? What is OPERON comprising of? So, Mr. Isa Nasser, how many things does it comprise of?
Promoter, operator, and structural gene.
Wow. So 5 things. So I can say this, that LAC OPERON has 3 structural genes under one operator. But this is not all about the LAC OPERON. This I want to make it very clear. That the OPERON is all that is highlighted. So if I ask you, promoter is part of OPERON, yes or no? Yes, no? Well done. ये reason है ना, ये तो बहुत important है, ये वाला, because this is where dash binds. Wow. And transcription starts. RNA polymerase binding site. And other things also happen here. हमने पढ़ा था decision that where should transcription begin. And also that strand selection. ये बहुत important बात है जो पढ़ा. Of DNA, which strand is transcribed. लेकिन आपको पता है, in this case, ZYA, थोड़ा out of course है, but in this case, the coding is on the same side. नहीं समझे? लोग जब DNA होता है, उस्मान भी हमारे साथ कैमरा ऑन कर. जब आपको पता है कि when a gene wants to show itself, so there are 2 strands, no? So one acts as template. The one that codes for mRNA. One guides the sense. So since there are 3, Here in lac operon, there are 3 genes. So for RNA polymerase, it's not possible to switch from this strand to this strand and this strand. So these 3 genes are arranged in a way that तीनों का जो कोडिंग पार्ट है ना वो एक साइड पे होगा. आप समझिए मैं कहता हूँ the tablet stand, yes, yeah. Did you get my point? But it was out of course anyway. But just in a way, okay, look, RNA polymerase will carry out transcription. So when RNA polymerase uses this strand as a template to make mRNA, so the same, this side of strand will be template for this as well. It's not that for this gene, this is acting as template, and for Z, this is acting as template. We are doing all about lack of one. मैं चाह रहा हूँ हमारा इस after this class we feel comfortable about this. इसलिए मैं थोड़ा सा तो हम slow लेके जाए. आपको समझ आ रही है जो बात है? So the first thing is what lack of one comprise of dash things. 5, nice. Yes, promoter where RNA polymerase binds. Operator where dash binds. Can you guess? Wow. Operator where a regulator binds, है ना? Regulated protein. Della is a good example. Miss Della. So operator where?
So this lac operon is eukaryote or prokaryote? Is this prokaryote or eukaryote?
This is in prokaryotes because in eukaryotes every gene has its own operator and promoter. But in prokaryotic DNA is very short, na? Sorry, beta? जी sure वो इसी पार्ट का वो इस चैप्टर में भी है. तो आप बताए कि where regulatory protein binds या आप कह लो कि DALA binds या where DALA is not the only regulatory protein binding. सही ना? So any question so far? So now what happens is ये basically lac operon is under the heading of a topic. You know what's the topic? Control of gene expression. Is that? So ये हम पढ़ें gene expression and prokaryotes with example to lac operon. Excuse me. More you need to know about this is what do these 3 codes for? So now lac Z code for what? माशाल्लाह ये अच्छा कभी भी याद नहीं होते. Oops.
That's for the hydrolysis of lac.
You know these names are in our course.
Yeah.
ये every year हम इसको थोड़ा सा डिच कराते हैं. ठीक है, अभी मुझे मैसेज आया था शहीर का, a student from another school. तो उसने कहा this is in the paper and they have asked you about the role of the.
Yeah.
है ना?
Yeah.
So this one is galactosidase. Yeah, you may call it beta galactosidase. So this codes for this.
Yeah.
And this one for what?
Lactose.
Permease, yeah. Mashallah, trans acetylase. You can say acetylase as well. So what shall this do? It will break down lactose into glucose and galactose. This helps absorb this.
Yeah, increases the permeability.
Yeah, and this helps in respiration of. इसका पता नहीं क्या function है. But you need to know the names at least. So what's important? By the way, यार थोड़ा सा break करते हैं. And may I ask you, what kind of enzymes are these 3? Inducible or repressible? Inducible. What are inducible ones? Which? Yes. बहुत अच्छा आपको आता है. Literally आप क्यों इसमें परेशान हैं आप लोग? Inducible enzymes are the ones which can be made by the presence of their substrate. And repressible are the ones which can be stopped by a repressor. देखें, like आप इसको it's only about the language how you define them. Inducible enzymes are the ones which are made only when their substrate is available. Yeah, who is making is induced by their substrate. And repressible are the ones which remain active every time, but sometimes you stop them.
Stop them
By repressor.
by repressing.
Yeah, by repressor. But if that repressor, मैंने तो पहले जब हमने पढ़ा था हमने ये कहा था कि अगर repression खत्म होती है substrate से तो वो inducible है. अगर सबसेट से नहीं होती वो रिप्रेसिव। मतलब देखें एक बात है मिस, अगर एक जो हमने जिब्रेलिन वाला पढ़ा था, तो याद है जिब्रेलिन क्या करता है, जिब्रेलिन गोज एंड बाइंड्स टू रिसेप्टर, जो तो सीड जर्मिनेशन में, और रिसेप्टर के साथ फिर एक एंजाइम अटैच है दैट गेट्स एक्टिवेटेड व्हिच ब्रेक्स डाउन डाला, तो फिर क्या होता है, एमाइलेज बन गया। अब देयर वाज नो स्टार्च, बट एमाइलेज का सबस्ट्रेट तो स्टार्च है ना, स्टार्च नहीं था बट एमाइलेज एक्टिवेट हो गया in the absence of starch. Why? Because you remove the repressor. So repressible वो होते हैं which can be which inducible are the ones which are active only when substrate is available. And then direct opposite to this definition is what? Repressible are the ones which are active all the time. Yeah, which are active even when the substrate is not available. Yeah, whose activity depends on repressor. Amylase. जो एमाइलेज था ना सीड जर्मिनेशन में, आपको याद है जिब्रेलिन सीड एब्जॉर्ब्स वाटर, फिर वो क्या करता था, जिब्रेलिन रिलीज करता था एम्ब्रियो। डू यू रिमेंबर दिस एम्ब्रियो पार्ट ऑफ सीड? ये बनाओ डायग्राम या ऐसा, ओके अभी आगे चलो बनाते हैं। तो एम्ब्रियो विल रिलीज जिब्रेलिन, विल गो टू एल्यूरोन लेयर, तो जिब्रेलिन वहां जाके बाइंड्स टू इट्स रिसेप्टर, देन एन एंजाइम गेट्स एक्टिवेटेड दैट ब्रेक्स डाउन डाला, because Dala was holding PIF. Eukaryote ना, seed is in eukaryotes. Seed कहाँ पे होता है बेटा? Eukaryotes. तो eukaryotes में असल में transcription factors नहीं है like PIF. Dala will break down, will get destroyed, PIF is freed, goes and binds to promoter and eukaryotes. तो फिर gene expression शुरू हो जाता है। इसका मतलब क्या हुआ कि एमाइलेज बना सीड में, एमाइलेज जो बना है तब बना है जब आपने डाला को ब्रेक किया। Yes or no? तो amylase को आप inducible कहेंगे? Because amylase is not made in response to starch. Amylase is made when DALA is broken down. इसका मतलब amylase नहीं बन रहा था उस वक्त से रोका था DALA ने, DALA को तोड़ो बन जाएगा, तो repressible है। यानी whose activity doesn't depend on substrate, it depends on repressor। लेकिन आप सोचेंगे यहाँ भी तो repressor यूज होता है, इसलिए हम यहाँ repressor की जगह उसे रेगुलेटरी का वर्ड कह देते हैं, सेम थिंग रेगुलेटरी प्रोटीन डाला। लेकिन इधर क्या होता है कि डाला रिमूव होगा व्हेन लैक्टोज प्रेजेंट, इसलिए देयर आर मल्टीपल लैंग्वेजेस यू कैन से, जैसे होता है ना केमिस्ट्री में मिक्सचर को आप डिफरेंट तरह के डिफाइन करते हैं ना, नो फिक्स प्रोपोर्शन, कंपोनेंट्स कैन बी सेपरेटेड, दे डोंट लूज द फीचर, इस तरह। तो इंड्यूसिबल रिप्रेसिबल को आप कह सकते हैं हुज एक्टिविटी डिपेंड्स ऑन द रिप्रेसर, यस और नो? या फिर आपको कि द वन व्हिच रिमेंस एक्टिव एवरी टाइम अनलेस रिप्रेस्ड, और inducible क्या है, the ones which are active only when substrate is present। नहीं ऐसी, चलो लेट्स कोई बात आगे चलते क्यों और पढ़ेगी। एक मैं आपको एग्जांपल बताता हूँ, मैं अभी डायग्राम बनाता हूँ, you'll get to know। चले ओके लेट्स हम एक डायग्राम बनाते, पहले ये पढ़ लेते हैं जरा इसको, इंड्यूसिबल रिप्रेसिबल को भी छोड़ो, मुझे बताएं डू यू अंडरस्टैंड द फंक्शनिंग ऑफ लैक ऑपरॉन? तो जो हो गया इसको हम पहले कंफर्म करते हैं फिर आगे
The operator is where the regulatory protein binds
yes
and prevents the RNA polymerase from being
binds to its promoter। या अगर आप ये वर्ड नहीं कहते तो आप यू मस्ट बी थिंकिंग कि प्रोमोटर तो फ्री है ना, तो यू कैन से कि प्रोमोटर से बाइंड करेगा, उसने आगे राइट भी तो करना है, टेस्ट बीज फॉर ट्रांसक्रिप्शन। तो आप कह लो ऑपरेटर इज वेयर डेला रिमेन्स बाउंड, डेला गेट्स bound and prevents RNA polymerase bind to promoter। अगर आप ये देखो डेला यूं होता है ना, यूं, दिस इज डेला, so if you want to say the bind stops RNA polymerase binding to promoter, that also is nice। If not, the other word is stops RNA polymerase from transcribing the, because promoter is on the left of operator, look RNA polymerase will bind to promoter, but in the middle of promoter and structural genes is the operator region, so RNA polymerase won't ride, cannot reach these genes। तो आप इसके मुझे जो समझ आ रही है, आप यही सोच रहे हैं ना कि if DALA is bound to operator, promoter is free, तो why RNA polymerase doesn't bind here? तो एक तो ये कि some part of RNA polymerase promoter is also engaged by DALA. But if you don't want to say, you say this, okay, DALA binds to operator and stops RNA polymerase transcribe the structural genes. Operators middle में. अच्छा, अब इस बार बेटा मुझे बताया कि LAC, Z, Y and A. These three code for enzymes. Yes or no? Okay. So those enzymes can be made only when dash is present. And what is lactose? Their substrate. So these enzymes are called inducible enzymes. So inducible enzymes are the ones which can be made only when their dash is present. Yeah. And like in chemistry, we say inductive effect. जी जी. अभी वो रिप्रेसिबल हम पढ़ते हैं एग्जाम्पल से ना मैं आपको एक चीज बताता हूँ फिर आप बिल्कुल क्लियर हो जाएंगे. जी जी. There is another definition parallel structural protein and regulatory protein है, वो भी इसमें देखेंगे. उस्मान भाई, do you understand it? उस्मान मुस्तफा. I got 50,000 which inducible are the ones which better can be made when they dash. Yeah. Done?
Yeah.
Yes. Acha, in the meanwhile, beta, can you please tell me one more thing? These three are proteins. Are they proteins or not? These are enzymes protein or them? So these are proteins. Will you call them structural proteins or regulatory proteins?
Which ones?
Structural proteins or regulatory proteins? Yeah, structural genes or regulatory gene. I mean the gene of these. Will you call them structural or regulatory? Okay, I'll say again. Structural genes are the ones which code for proteins which are of the use of to the cell.
There's the LAC, ZYA, they are structural.
They are structural proteins.
And the beta galactosidase is big deal.
No, these all are. Look, the beta ZYA are structural genes, and the enzyme they produce are inducible. You can call these to be structural proteins as well. What are structural genes? Ones which code for proteins which are of direct use to the cells. But regulatory genes कौन से हैं? Which code for protein? ये तो आपको पता है ना, gene codes for a protein. Yes or no? So what are regulatory genes? Which code for protein which control regulate the expression of other genes. For example, gene that makes DALA. What is that? Regulatory. Yeah. और वो इधर ही होते हैं, you know, इधर ही होते हैं. For example, if you extend this DNA ahead, there will be some regulatory gene और ये उसका operator, ये उसका promoter, सारा कुछ. ठीक है, आप सोचेंगे regulatory gene को control कैसे करते हैं, ठीक है ना, वो हमारे कोर्स में नहीं. But the important thing is कि are we done with structural and regulatory genes?
So what's the definition of structural?
Structural genes is genes which code for proteins which are of direct use to the cell. And regulatory genes are genes which code for proteins which regulate the expression of other genes. Now, example. Structural genes का example है gene of this LAC Z, LAC Y and LAC A. Because all three genes code for proteins which are of direct use to the cell. These proteins help cells perform some function. They don't control the expression of the gene. But the gene that codes for DALA is regulatory gene. Gene of amylase is structural gene. और वैसे amylase enzyme क्या है? Inducible या repressible? Wow, जबरदस्त.
The genes that structural genes are genes that code for
Proteins which are of direct use to the cell. अच्छा, ओके ओके, ठीक है. what you can do is you can please type if something bothers you, because today we were doing inheritance assignment ना, हमने कहा ये कुछ लैक ऑफ का टॉपिक देखते हैं, तो आपने अगर कोई चीज ऑन कर दिया आपने मैसेज टाइप करना, ठीक है, ताकि आई वांट कि आज आप इनहेरिटेंस के, यू नो इनहेरिटेंस वी आर डन विद दिस बिफोर ना, तो व्हाट आई बिलीव इज कि आप थोड़ा सा आप प्लीज इफ यू पुट योरसेल्फ इन ट्रबल टुडे एंड गो थ्रू ऑल द असाइनमेंट, है ना, सो वी सी द टफ वंस, बिकॉज सॉरी, हाँ, regulatory genes are genes which code for proteins that regulate the expression of other genes. Now, beta, examples of structural gene कौन सी होंगी? LAC, yeah, these are structural gene. उसके आगे आप लिखते हैं for example LAC Z Y A. और examples of regulatory gene क्या होगा? Gene of Dalla. अच्छा, अब हम ईसा भाई एक बात, look, this is not yet clear ना, inducible and repressible. चलो inducible तो आप मुझे बता सकते हैं, what are inducible enzymes? Look, what is, I hope I'm not confusing things. Look, there are two types of genes and two types of enzymes. So genes are structural and regulatory genes. Enzymes are inducible and repressible. So I hope we are done with structural and regulatory genes. अब आप मुझे बताएं, gene that codes for hemoglobin, is that structural or regulatory? Wow, है ना? Because right, bhai, because hemoglobin doesn't control the expression of other genes. Hemoglobin has separate job, like it oxygen virus. अच्छा, मुझे बताएं gene that codes for PIF, क्योंकि PIF भी एक प्रोटीन है, फाइटोक्रोम, नहीं, वो तो एंजाइम्स है ना, आई एम टॉकिंग अबाउट जीन्स, रेगुलेटरी जीन. तो शी नोज के जीन दैट कोड्स फॉर PIF ना, तो PIF क्या करता है? PIF एक्सप्रेशन ऑफ जीन को कंट्रोल करता है यूकैरियोट्स. तो इसका मतलब PIF इज अ रेगुलेटरी प्रोटीन, सही है ना? All the genes which code for transcription factors are dash genes. Now, what are transcription factors? Factor means a small protein. देखो फैक्टर का नाम पता है, फैक्टर 8, ये चैप्टर में फैक्टर 8 क्लॉटिंग वाला, तो वो जो फैक्टर 8 है इज अ प्रोटीन व्हिच हेल्प्स इन ब्लड क्लॉटिंग. तो मुझे लगता है फैक्टर 8 का जो जीन है, इज दैट रेगुलेटरी जीन या स्ट्रक्चरल जीन? It helps you clot, helps us clot the blood after the wound. Structure. So factor 8, ठीक है ना? तो फैक्टर क्या होता है? प्रोटीन. So clotting factor is the protein which helps blood clotting. But transcription factors are protein which help which control gene expression. So genes which code for transcription factors are dash genes, structurally regulatory. Genes which code for genes which code for transcription factors are regulatory gene or structural genes? No problem. What are transcription factors? आप बताइए, transcription factors क्या होते हैं? Yeah, factor, word factor means protein. छोटे प्रोटीन को फैक्टर कहते हैं. तो transcription factors are proteins like which control gene expression. The good example of transcription factors is PIF. Yeah. Factors are many. Some factors may determine our gender, you know. हम जो मेल और फीमेल हैं, हाँ, this also depends on transcription. There are some proteins other than being X and Y. You cannot be male if some proteins are not there. Transcription factors have lot of roles to play. ये आपने बुक में भी लिखा है, gender determination भी लिखा है. So my question is that PIF is one of the transcription factors. What are transcription factors? Small proteins which control the transcription, मतलब which control expression of genes. तो ईसा भाई वो जो ट्रांसक्रिप्शन फैक्टर्स हैं, control transcription factors are small proteins which control the transcription या gene expression. yes or no? बेटा आपको पता है ना, जीन जब भी शो होएगा वो ट्रांसक्रिप्शन करेगा ना पहले. तो genes which code for transcription factors are dash genes. Like regulate, ये easy हो गया. There's still problem.
So DELA is transcription? Is coded for a transcription? No.
Good. DELA is the regulatory protein, but we don't call it transcription factor because जो है ना DELA and prokaryotes, it directly binds to operator and stops the gene expression. So we call it repressor over there. So transcription factor is a specific word for eukaryotes. So since it has a role in prokaryotes as well, otherwise it has every reason to be called as transcription factor. One more thing, DELA is a little bigger as well. Transcription factors are shorter proteins. The word factor is for proteins which are made by few amines. So, miss, are you confused here linking with this? Why DELA is not called transcription factor? No, why DELA is called, should be called transcription factor? Because it controls gene expression, right? But, and why it is the transcription factor? Because it controls gene expression in eukaryotes. Anything that controls gene expression in eukaryotes is what? Transcription factor. So इसलिए ऐसे dialogue आप कह सकते हैं. But why it is not? Because number one, it controls gene expression in prokaryotes also. And secondly, that in eukaryotes, this does not bind to promoter or RNA polymerase. Another feature of transcription factors is that they either bind to promoter or RNA polymerase. Let me write this again. मैं इसको लिखता हूँ. How much of that? What is transcription factor? वाह, देखें, एक तो इसमें यह कि स्मॉल प्रोटीन, व्हेन यू से फैक्टर मीन स्मॉल प्रोटीन, जैसे क्लॉटिंग फैक्टर होता है, स्मॉल प्रोटीन, ठीक है, कंट्रोल ट्रांसक्रिप्शन या जीन एक्सप्रेशन एंड व्हाट एंड यूकैरियोट्स ओनली, एंड थर्ड they either bind to promoter or RNA polymerase. Very good. This is how they control the gene. If they are not bound to promoter, gene will not be shown. So now, by this all introduction, you will not believe that DELA is a transcription factor. Let's see what DELA does. Then after that, we'll go back. The why DELA is not a transcription factor. अच्छा gentlemen, मुझे ये बताइए जल्दी से कि inducible enzymes are the ones whose which can be made when their substrate is there. Yes. अब देखो मैं एक एग्जांपल बताता हूँ. Seed. This is हमारे कोर्स में ना seed germination. ये माइक्रोपाइल है, छोटा सा पोर, barley seed. And you know what is that? The layer. Usman bhai, can you type for me the outermost layer of seed? Aileron. And this one is embryo. You know it, mashallah. Nice. So this is aileron layer. जल्दी से हम इसको करते हैं. Aileron. This is endosperm, which is food reserve. ईसा भाई बहुत important है. For example, this is endosperm is a food reserve. For example, starch. जो मैं आपसे कह रहा था ना लारे कि ये अभी हम पढ़ेंगे, आपको समझ आ जाएगी. Starch. अब देखिए, starch is the substrate of what? Amylase. But amylase is not expressed in unfavorable conditions. So what is amylase? Inducible, repressible. Repressible. Even if its substrate is there. Which is substrate for amylase? अब देखो, तो seed does not germinate. अगर आप seed को germination condition proper दे दें, राइम भाई, अगर seed की condition germination proper होंगी, so there is starch also present. So don't you think amylase will keep making? और plant grow करता जाएगा. इसलिए तो जब condition favorable हो, तो amylase यूं break down करता है और वो maltose बनाता है, glucose बनती है, embryo बनता है, radical promule and plants grow like anything. तो अगर conditions reasonable होंगी, suitable moisture, air, temperature, तो amylase will keep forming. So why is amylase called repressible? अब वो आपको समझ आ गया होगा बात कि जो मैं कहता है हर वक्त बनते रहते हैं.
Does it only function when?
That they repressible are the ones whose activity can be repressed, otherwise they continue. So why amylase making was repressed? By drought. It was dry, या there was like, जैसे सीड बैंक में क्या करते हैं, सीड बैंक में बहुत लो टेम्परेचर रखते हैं.
So when gibberellin binds, then amylase.
Sorry.
सीड बैंक में फिर हमारी एक्टिविटी नहीं होती.
एक्टिविटी नहीं होती क्योंकि आपने उसको रिप्रेस कर दिया. आप क्या कह रहे हैं?
So when gibberellin binds to the receptors in the neuron there,
वो भी पढ़
that allows amylase.
वो भी पढ़ते हैं, हाँ. So the idea, first thing is that amylase is the example of dash enzyme. Repressive. That look, even in spite of the fact that its substrate is there, yes, it is not being shown. But when water is present, हमने पढ़ा था, there are some facts we need to recall. कि अगर water, water is a condition for germination. Water absorbed by embryo. इसको मैं वर्ड्स लिख देता हूँ, सही? Embryo secretes gibberellin. अच्छा पता है, gibberellin is not scientific name. Scientific name is gibberellic acid. It's like GA1, GA2, GA3 इस तरह होते हैं. ये हमारे कोर्स में नहीं है, but questions में वो पूछते हैं. किस तरह? They ask you, look, a plant has gibberellin genes, is making gibberellin. No, sorry. अगर आप gibberellin spray करो, फिर भी growth नहीं हो रही, तो maybe there are no receptor for it. या maybe you are spraying inactive form. You understand है ना? तो अब देखें gibberellin बना, gibberellin goes to aileron layer. अच्छा, now the story starts very, very interesting. What happens to aileron layer? See, gibberellin binds to its, ये मैं कह रहा था, binds to receptor, फिर enzyme gets activated, yes or no? Enzyme is attached on the inside of it. So enzyme gets activated or released from receptor. And this will destroy what? Dala. And Dala was holding what? PIF. So PIF is free. Isa, what is PIF? A dash factor. Yeah. So PIF gets freed. And where does this go? Where does PIF go? Goes and binds to either promoter of amylase or RNA polymerase. Yes. This is how transcription factors help transcription. So amylase will be expressed. Any problems so far? आपने इतना सब पढ़ा है. If you zoom it out. Sorry? ये यूकेरियोटिक. मतलब यूकेरियोटिक में फर्क है कि यूकेरियोट्स में अब ये उस्मान कुछ कर रहे हैं. Eukaryotes में its transcription factors which control the expression. Usman, can you take the picture? His mic doesn't work. Usman, please take the picture of this. I hope you are done with.
So gibberellin bind करता है receptors से, तो वो एक enzyme को activate करता है जो break down करता है DALA को.
और PIF free हो गया. PIF will bind to promoters of amylase. Amylase express हो जाएगा. इसलिए कहते हैं. अब आपको पता है inheritance में इसकी debates और भी बहुत complex. There are complex debates. किस तरह कि how can you make plants grow taller even if they are genetically dwarf? तो जो genetically dwarf plants है ना, वो क्या करते हैं? There are two genes, two alleles of a gene of height, for example. So one जो dominant है ना, that is making a GA1, active form of gibberellin, and जो recessive है, that is not making gibberellin. Yeah, if it is making, it is making inactive form. सुन रहे हैं? So if you have two recessive, if a plant has two recessive alleles of a gene which is making wrong gibberellin, so don't you think that plant will be short at all? Right? By plant height gene has two alleles. One codes for active gibberellin, the one codes for non-active. तो अगर आप homozygous recessive है, तो उनके या तो जिब्रेलिन बना ही नहीं गए या इनएक्टिव बना, समझ रहे हो?
Either they are,
Not making gibberellin or they are making inactive gibberellin.
or they're making inactive.
Inactive gibberellin, because gibberellin has like GA1, 2, 3 different forms of gibberellin. तो वो inactive, मैं आपको कॉल करता हूँ. तो you know they like अगर आप स्प्रे करेंगे जिब्रेलिन का तो क्या होगा, वही जिब्रेलिन will go and bind to its receptors, है ना?
जो dominant वाली है वो jubilee active form वाली है.
हाँ, मतलब height gene has two alleles, right? For example, which one? Like one allele, dominant allele makes active form of gibberellin, G1, and recessive makes inactive form. Yeah, doesn't make the gibberellin, है ना? Not necessarily. Sometimes dwarf plants इसलिए भी होते हैं कि उनमें receptors for gibberellin नहीं होते. ये सिलेबस में नहीं है. You know, a question was there कि you sprayed gibberellin, बेटा investigation है. ऐसा भाई gibberellin spray किया, one group of plants, the other group of plants, one grew taller, the other couldn't. Because thank you Swan bhai, the ones which grew taller are